All right, thank you. Yeah, let's dive right in. So if you think of tissues, at least we at Vizgen, we like to compare them to maps. And similar to a map, let's say a digital map, Google Maps on your phone, you can access the information in that map at different layers, right? You can zoom in, zoom out, and you will access different layers of information in the map. And depending on the question you have, different layers of information will be important. For example, you want to zoom out if you're interested in borders between countries. You want to zoom in if you want to know all the streets in your immediate neighborhood. And similarly, then in tissue biology, you may need different techniques to access different layers of information, different layers of magnification, to get the information you need for your biological question. And at Vizgen, we offer two products that access different layers of information, and I'll present those two to you today and what you can do with them. So starting with H&E, which you all know, that's obviously, of course, a very important technique already by itself, that you can get rid of a lot of information already just from that assay. But you may want to go one step further and ask the question, which cells express which proteins, right? And for example, you want to distinguish cytotoxic T cells from T helper cells. And maybe you want to measure the distance between cytotoxic T cells and tumor cells, whether they engage, and so on and so forth. For that, we offer the in situ plex assay, which is a spatial proteomics assay where you can visualize up to 12 proteins on your slide using that assay. Then you may want to dig even deeper, zoom even further into your tissue, and maybe be able to distinguish more subtle subtypes. Like, what different cell subtypes do I have within my tumor, and how do they differ in gene expression? And that is a question you can answer with the MERFISH technology that allows you to profile up to 1,000 transcripts to 1,000 markers spatially. And so I'll speak about these two techniques and how we implemented them and how you can use them. And then in the last part of the talk, I'll give you an overview how you can actually combine these two measurements and to get even more information, valuable information from the tissue. Let's start with the proteins. So we sell this under the name of in situ plex, and essentially it's a kit that you can use on your slides. And I'll guide you through the journey from sample to analysis, all the way then to biological insights that you can generate using that technique. So I'll go through the protocol briefly. So there's the DWAX and antigen retrieval step, and then you incubate the sample with the antibodies, and that can be either 4, 8, or 12 antibodies at the same time. You can buy these antibodies from us. They are conjugated so that they work with the in situ plex technology. And you can go to our website and choose from a menu of 24 pre-developed markers that are developed, tested, and verified. But you can also make a request and ask us to develop an antibody for you. So if the antibody you are working with, the biomarker you're interested in, is not part of that panel, we can develop that marker and make it available in the context of an in situ plex panel. So we will also do the necessary validation steps so that you can be sure that it really works as expected, even in the context of ISP. So once you did the antibody incubation, the next step is then signal amplification, which is specific to our assay, and that uses DNA barcoding technology where the barcode gets amplified. And this is very important because it will allow you to get really good signal-to-noise ratio, even in challenging tissues where you have high autofluorescence, for example, and you'll be able to capture at the same time low expressors and high expressors because it's a linear amplification system. So that is important. Then in the next step, you incubate with a probe mix that also comes with a kit, where it's again a DNA sequence attached to a fluorophore that will then specifically bind your antibody so that you will get a specific antibody fluorophore pair, which then allows you to image always 4 biomarkers at the time at the microscope. Why are we only imaging 4 biomarkers at the time? The advantage of that is that you can use the microscope, the slide scanner that you already have in your lab, because we just require this microscope to have the 4 classical filter sets, FIT, CIT, Psi5, Psi7, which usually comes with fluorescent slide scanners. So you don't have to buy a fancy new microscope. You can just use the microscope you already have. And also the other advantage is that we don't rely on spectral unmixing, so we can just take the raw data as it comes from the microscope, because optically the signal is well separated. Then, of course, in the case of an 8-plex, if you want to visualize 8 biomarkers, you just do a signal exchange and then image again the other 4 antibodies. So that gives you then for each sample 2 images, which each containing the information of 4 biomarkers. That means that in the next step, you have to register these 2 images to get the full information for every sample. And we can do that, and we developed a software for that is called AltiStack AI. And as a Superplex user, you will get access to that software. And it does 2 things very well. It does the correlation very well at micron accuracy, which is, of course, critical to do proper cell phenotyping downstream. But also it scales very well. So where other solutions require some manual intervention to do this registration, here it's really a one-click solution. It works in the cloud and is very efficient, so you can easily stain 20, 30, 50 samples in a few hours, stack them. And so that is really great, because in the end, what you will end up with then is an image like this, which is a colorful image, lots of cells, and then for each of the cell, based on the panel you used, you know which protein it expresses. And then, for example, naive regulatory T cell, you would expect these 3 markers to be expressed, CD3, CD4, FOXP3. Now, of course, the next thing you have to do is to phenotype all of these millions of cells to be sure what proteins they actually express. And there again, you want the software that does this to be able to do 2 things. It has to work very accurately, robustly across tissue conditions. And second, it also has to scale well, because it's millions of cells. We always image the full slide, not small field of views. And you might have 50, 60, 100 samples in your cohort. So it has to scale also across larger numbers of samples. And again, we developed our own solution to do this, AltiAnalyzer AI, which uses state-of-the-art deep learning models that are performing object detection similarly to self-driving car, who makes the decision to distinguish between a pedestrian and a street sign. In our case, we train the model to distinguish a CD4 positive from a CD4 negative cell. And we do this specifically for all of our core markers. So for every marker, we have a dedicated deep learning model. And that has the advantage that the model doesn't only look at the signal intensity, it really takes also the local context. So when deciding for every cell, it looks around, so to speak, and then it really learns, or we train it that it learns the expected shape and staining pattern for all of these markers. And that makes it much more robust to things that can happen in the tissue, autofluorescence, out of focus, and so on. And so we have very good experience with that. And that together then gets integrated in a flexible framework where you can also use other segmentation methods, not only object detection, but we also do region segmentation where we delimitate cytokeratin positive regions from cytokeratin negative regions to do a tumor stroma segmentation. And it can also include other data types, such as manual annotations drawn by pathologists, for example, so that you only get the analysis for the areas in the sample that you're interested in. So now we went from staining the sample, imaging it, stacking the different rounds, to analysis, which makes you then end up with a long list of cells. And for every cell, you know which proteins it expresses and also where in the sample that cell sits. And now you can start to ask the really interesting questions, because now you can ask questions like, okay, across the samples in my cohort, is there a difference between the distance between tumor cells and cytotoxic T cells? And maybe does that difference correlate with the treatment a subset of patients in that cohort received? Things like that. That's what we call biological insights. And yeah, that's something you can do with this type of data. So this is just an example, the collaboration we did with Frieda Nössner's lab in Munich. And here on the left side, you can imagine that the yellow cell would be a tumor cell and the magenta cell would be an immune cell, say a cytotoxic T cell. And then one score that we implemented is calculating the average distance between a tumor cell and its nearest cytotoxic T cell, and doing that across the millions of cells that we have in the tissue. And then you can do this for one cell pair, but you can also extend the analysis then to other cell pairs which results then in a plot like on the right, where you can stratify the patients in your cohort based on their, let's say, interaction fingerprint and how they differ in cell-pair interactions, which gives you then already a valuable spatial result. This is just one example of an algorithm we can implement. There are others also used in the literature that have been also used and correlated to clinical outcomes. And the importance here is that we can implement all of those with minimal effort because of the flexibility of our solution. The other example I want to briefly show you is that you can also leverage the H&E component. So after an ISP run, so after doing the last fluorescent staining round, you can strip off the fluorophores and then perform an additional round of H&E staining at the end so that you get the same slide also stained with H&E. You can then register the H&E on top of the ISP so that you get the same information for the slide, or the H&E information and the ASP information for the same slide. And here, this was an example in renal cell carcinoma where we had tumors that didn't really express or had just very low expressed cytokeratin levels. And that is a marker we usually like to use for tumor stroma segmentation, so we couldn't do that here. But then we could just go to the H&E signal, train an algorithm there so that we rely on H&E for tumor stroma segmentation. And that is just one example how to combine these two measurements. But the important thing is that it gives you a lot of advantages when you have H&E corresponding to the same slide where you also have the ISP, the fluorescent data. Okay, so that was proteins. Now switching gears a bit, zooming in further into RNA space. So that is what you can see on the very right. Spatial RNA expressions. You might also have heard of spatial transcriptomics. And the technique we use and implemented now as a product is MERFISH, that was originally developed at Shuai Zhuang's lab at Harvard. And yeah, MERFISH stands for multiplexed aura-robust fluorescence in situ hybridization. And you might be familiar with single molecule FISH, which is very related, which is basically a technique that allows you to visualize individual transcripts with high sensitivity and specificity. But the trick now MERFISH is doing is that you can scale this method to hundreds and even thousands of transcripts. How does it work? I won't give you all the details, but this is all published. So if you're interested, go to these papers. But in the end, what it does is assigns a binary sequence to a transcript. So you do sequential imaging, and then each transcript will be off. So there will be no signal in some rounds. And in other rounds, there will be an on signal. So you will see a dot. And the combination of on and off events then gives you the transcript ID. And that is a bit similar to a Morse alphabet. We also have a binary sequence that encodes a letter or word. And here, the binary sequence encodes the transcript ID. The good news is that you don't have to worry about this complicated decoding and coding because it's done automatically on the machine. So MERScope is the product we sell and we have developed, and that uses MERFISH technology, but it comes as a box that you can use and where you don't have to worry about a lot of the technical implementations. The only thing you have to do is the sample preparation. That, of course, is specific for each and every tissue, but we, of course, have a lot of experience and can provide you with protocols for your tissue. And then you just put the sample on the slide and then put it in the MERScope, and then everything will run automatically. So the microscope is optimized to image at very high resolution, which gives you, yeah, very good and accurate cell profiling then, because you can visualize the spots very nicely. And then it will also handle the chemistry automatically. So the sequential imaging and the application of the different probes, that is done automatically with microfluidics. And also the decoding and basically mapping these binary on-off events to a transcript, that is also done automatically. You don't have to worry about it. We are actively developing that product still, and recently we launched the MERFISH 2.0 chemistry, which is a new chemistry that is available for all new but also existing MERScope customers that increases the sensitivity of RNA detection in challenging tissues with low-quality RNA, such as FFPE tissues. And here, this is just an example where we measure the sensitivity by transcripts per area, and you can see a drastic increase for the MERFISH 2.0 chemistry, and customers were also really happy about it. So MERFISH and the instrument, the MERScope, has been used in a lot of publications by different groups, companies, and maybe more importantly, also across tissue types. So it's a very versatile method that works across organisms and tissues. So we have very lot of experience in the mouse, fresh frozen or fixed mouse brain, but also other tissues, even whole mouse embryos, as you can see here. Human tissue, of course, tissue microarrays, also plants. So that's something that's really interesting about this method. Okay, so I told you about proteins. I told you about gene expression. How can we combine the two? Now, being a company that offers both assays, can we combine these two assays? And there we did a proof of concept experiment where we generated matched multi-omic data, and I'll explain how we did that. So we took serial sections from two different indications, and yeah, the sections were adjacent, and then one section goes to the MERScope and is profiled with the MERFISH 2.0 chemistry, and the other section is subjected to an in situ plex panel, 12 plex in this case. And there we run our custom analysis that I introduced before, deep learning-based cell phenotyping, and also region segmentation, so that we outline the tumor using the cytokeratin expression. And then again, we set out to combine this morphological information that is based on the in situ plex protein data with MERFISH data. And we did two experiments, using cytokeratin as a region segmentation marker, applying it to MERFISH data. I only have time to speak about this one. If you're interested in the other one, which is the correlation of gene expression and how certain gene expression modules are actually correlated to cytokeratin as measured in the protein space, please go to this link. You can download the posters there, because this is all based on work we released during SITC this year. And there you can download the posters. I'll guide you through the second experiment, where we discovered subcell types or subtypes of cells within the tumor. And the tumor was basically delimited by cytokeratin expression. So really using the two modalities, and that was a case of non-small cell lung cancer that we used here. So now it's getting a bit technical, but I'll try to guide you through it. So if you have these two adjacent sections, let's assume that the upper section is the ISP data, so the one that where we did protein imaging. And then for that one, we get basically the areas as polygons where cytokeratin is actually expressed, and that would be our tumor region. Now, then you have to align this to the MERFISH data, because again, these are coming from two adjacent slides. This is not from the same slide. There we use a tool that is called VALIS, that is open source. And we basically use the DAPI that is imaged in both techniques to do the co-registration, so that then in the end, for every cell in the MERFISH data set, we know its gene expression, so that we know which transcripts are expressed in that cell, but we also know whether that cell overlaps with the cytokeratin positive region, yes or no. And that is what is shown on the right then. Importantly, that's an important disclaimer, is that this is not single cell perfect, right? Because we are not speaking about the same slice. So there's a maximum distance of 10 microns between the cells. So this won't be perfect at the single cell level, but we generally assume that over these 10 microns, the overall tumor architecture is conserved, so that the cytokeratin identity of cells in the MERFISH data should be conserved across this distance. So this is how the spatial transcriptomics data look like. You can plot it in a lot of different ways. It's highly dimensional. Every dot here on the left is a cell, and in the left you see it in the spatial coordinates of that sample, and then on the right every dot is a cell again and colored by cell type, but there it is positioned in its gene expression space. So generally, if two cells are close to each other in that plot, that means that they are more similar in gene expression, whereas if they are far distant from each other, they are distinct in gene expression. So, for example, it is logical that tumor cells here in orange, they are far away from the immune cells that you see on the right, because they're transcriptionally distant, as expected, right? And the first exercise we do then, as I told you, is to transfer the cytokeratin mask to the MERSCOPE data so that we know for each cell whether it's within a cytokeratin positive region, yes or no. And then we did an additional stratification step where we look at all the cells within the cytokeratin positive regions and define it to be either a tumor core cell in the center of the tumor or a tumor edge cell, and that is if they are within a 15 micrometer band of the tumor. And that is something that really highlights the use of the protein here, because only the protein signal will give us the sharp bands that we need to do accurate band construction, so to say. So using the protein signal as anchor, we can integrate the gene expression data. And so having this compartmentalization, we can then ask interesting questions like, okay, if we have cells inside the tumor and at the edge, can we identify gene expression modules that are different between these two cell types? So do centers at the cell, do they have a different transcriptome than cells at the outside of the tumor? And that is then something we did here. I'll guide you through the plot on the right, where on the y-axis, it gives you the gene expression level relatively, and every dot is the cell again. And then on the x-axis, it gives you the spatial positioning of the cells relative to the border. And so blue cells are in that 50 micrometer band. And so by performing differential gene expression, we can then identify genes that are enriched in this blue band. And on the right, what you see is an example where the distribution is much more homogeneous and maybe even a bit enriched in the tumor, because you see many more orange dots than for the other, for the left plot. And now these are the top hits that came out in this differential gene expression analysis. MCM2 is a proliferation marker. The protein is involved in replication of DNA replication. And CDKN1A is a kinase that is involved in senescence and cell cycle arrest. So that makes sense that what we can learn from this is that the proliferation is happening at the outside of the tumor, whereas the tumor core is rather senescent. And so again, the goal here was not to discover new biology, but it is reassuring to us that we rediscover something that makes biologically sense. And importantly, that would be difficult or more difficult to get to when relying only on gene expression data, because you wouldn't have the protein signal to anchor your cells. Then you can zoom in even further in the MCM2 expression pattern and see, for example, that the expression level is specifically high around the tumor areas, so more yellow, and then map it back in gene expression space. And there you see that the cells having high expression of MCM2 are forming a subcluster within the tumor cells. And that is also interesting because it means that MCM2 is not a technical outlier that somehow cells in this tumor band express, but it's more likely to be part of a gene expression program because overall in gene expression space, cells having high MCM2 expression clustered together. This is for reference the tumor cells again. So it's the same positioning of cells, just the color code changes. And so all of this to say that here we did a proof of concept example where the integration of protein-derived tissue morphology, in this case we use cytokeratin, but it could also be another marker, with high-plex MERSCOPE spatial transcriptomics data improves our understanding of what is going on in the tumor. So again, we use cytokeratin here, but you could also think of another marker that gives you morphology-related features on the protein side that allow you then to eliminate your tissue better so that you can then do differential gene expression in the high-plex space in the MERSCOPE data. Okay, that brings me to the end. So that's what we offer at Vizgen. We have a spatial proteomics assay that is called in situplex. We have a spatial transcriptomics assay that is based on MERFISH, and we sell it together with the machine, a microscope that is called MERSCOPE. We recently released a new chemistry for FFPE tissues that works really well. And Importantly, I try to bring across that we deeply care about extracting the information from the images we produce with our techniques. So it's not only about pretty images. In the end, you want to learn something. And we have developed tools and software to really help you with that. And you will get access to these tools either as a user, or what you can also do is we provide also you with a services offering where you can send us samples. We will do the imaging, the staining and imaging, and then also the analysis. And there we can also customize the analysis to your needs. So if you're interested in that, or if you have a cool multimodal experiment in mind, please come talk to us at our booth 27 for more information on that. Feel free also to download the posters. Thank you for your attention, and I'm happy to take questions.